First of all, thank you to the few and the mighty that have managed to stay all the way into the third day of farm. I'm Paul Ash. I'm CEO of Vesigen. In the next 15 or so minutes, I look forward to telling you our story. It's a story of the possibilities of novel therapeutics unlocked by engineering this natural class of extracellular vesicles. So this audience probably well knows this fact, and if you didn't, you've certainly heard it in spades in the last couple of days. We have all kinds of ways to edit biology in the biopharma R&D community, whether it's base editing or prime editing or gene writing or protein effectors and degraders and RNA therapeutics. The list goes on and it seems to grow every day. But all these approaches get stuck at delivery. Whether it's the challenge of redosability, which plagues viral vectors, whether it's getting reliably to tissues outside the liver, which is difficult for LNPs. Manufacturing for all these things is very costly and complex. Staying under dose-limiting tox is a challenge. In short, the problem of delivery has not been cracked. Our vision at Vesigen is to get some of these advanced therapeutics delivered very efficiently to the very specific tissues and cell types within those tissues. So let me tell you on this slide a summary of our value proposition in delivery, and I'll go through each of these points in a little more detail. So I'll start on the top left in light blue, large cargo capacity. And by cargo, I specifically mean the little pictures on the left. So gene editors, this is a fully functioning RNP, so CRISPR-Cas plus the guide, plus potentially a deaminase. It could be just the protein piece, it can be just the RNA piece. And when I say large, we can fit up to 20 sort of standard issue CRISPR-Cas plus guide units in one vesicle. If it's something larger, like an adenine or a cytidine base editor, you're looking at 10 to 15. If it's one of the smaller nucleases, you can fit a lot more in. The middle panel, differentiated by a distribution. It's very hard to talk about delivery without talking about where you're going. You can see with the icons there some of the tissues that we can get to reliably: eye, lung, spleen, liver. The list is actually a lot longer. This is where we have the most data. And that's the natural tropism of this class of vesicles. We've also uncovered the ability to direct that tropism to get to other cell types of interest, which I'll speak to more. On the right, tunable functional delivery. So if you're in the world of LNPs or exosomes, the way these particles get introduced to the recipient cell is through a sort of a classic endocytic pathway. Whatever is able to escape from that endosomal trap gives you your therapeutic effect. What you see here is a membrane interaction, right? So envision the contents of the vesicle being dumped into the cytocol en masse, and you can add a lead sequence to get to the nucleus or compartment of choice. Bottom left, redosable. In the whole history of the company and all the experiments we've run, we haven't seen any immune response. No neutralizing antibodies, no TLR activation, no global transcription changes. All of this is probably likely due to the fact that we're talking about a natural vesicle which gets cleared by the natural clearance mechanisms of the body, easily paves the path for redosability. And on the bottom right, I've been impressed listening to some of the presentations over the past few days on how much of the presentation is dedicated to manufacturing. And this is probably not a surprise because manufacturing takes a lot of the resource of a company. Vesigen's pretty different here. Our process is quite straightforward. There are very few steps. They all start with standard off-the-shelf materials, and the unit ops are well known. So there's lots of CDMO capacity, and you can kind of easily contemplate a third-party strategy. So let me tell you on this page how the technology works, and then I'll go back into each of those attributes. So our vesicles are called ARMS, and that is not so much a nod to our hosts here. It's an acronym for ARDC1 mediated microvesicles. That is a mouthful, but it's a class of naturally existing vesicles that we can essentially co-opt to deliver some therapeutic payloads. We think co-opt is a better word than hijack. It all hinges on this protein called ARDC1, which is depicted here in this green triangle. If you take your producer cell line and append to ARDC1 a tether and the code for your protein of interest, the cell line will actually take ARDC1, take the payload, which is depicted as the blue oval, and drag it into the interluminal space of the vesicle. So the cell is actually doing the loading work for you. And as you see the vesicles in transit, See the green triangles, you see the blue payloads. By the time they get to the recipient cell side, you see that membrane interaction and kind of the emptying of the contents into the recipient cell side. So that's kind of how it works, and again, it hinges on the existence of this protein that's responsible for the biogenesis of these vesicles. So biodistribution. What you see on the left is a couple of different routes of administration: intraocular, intranasal, intravenous. We have a bunch more not depicted here, like intracerebroventricular, intrathecal. Again, this is where we have the most data. In the middle, you see the icons for the tissues we get to, and then the very specific cell types within those tissues. I'll pick a couple of examples. In the eye, if you do a subretinal administration, you can get to the retinal pigmented epithelium and photoreceptors with high fidelity. As you see from the pictures on the right, we've shown this in mice, we've shown this in mini pigs, and as of a couple weeks ago, we've shown this in NHPs. Robust translation across species. In the green, if you do an intranasal administration, you get to alveolar macrophages. And again, not macrophages as gobbling up cells, macrophages as an ability to deliver an edit. I'll also highlight in the last row there, in IV, when you get to the liver, hepatocytes are the target for LNPs, as you well know. So if you see any of these companies that are using LNPs, they are usually, almost always, addressing a disease whose pathology is resident in that one cell type, alpha-1 antitrypsin deficiency, et cetera. ARMs do not get taken up by hepatocytes. They get taken up by Kupffer cells and liver sinusoidal endothelial cells. LSECs are the one cell type in the body that produces factor VIII. So you can start to contemplate a curative solution for hemophilia instead of a very regular, almost daily introduction of enzymes. Or there's several AAV-based approaches, some of which have durability challenges. If you run into a durability challenge, you're stuck, right? Because you can't take a second dose. With this, you're, again, getting to the one cell type that's responsible for all the factor VIII production, and contemplate an edit which does a permanent fix, and if you don't have the dose exactly right, redosability, not an issue. So it opens up some very interesting possibilities. So here's a list of some of those diseases. Stargardt, there's a bunch of approaches to Stargardt out there. This one's a little different. So I'll contrast with a dual AAV strategy, which there's a few out there. AAV is complex, right? And 2AAV is that much more complex, but you need the dual strategy because you just don't have the cargo capacity. You also have to worry, in that case, about persistent expression of Cas9 in the eyes for the life of the patient. Right, so here, it's quite different approach. It's a transient delivery, right? So you can fit all your editor into one vesicle, you do the edit, the body's natural clearance mechanism clears it out quite quickly, and you have no persistent expression of your editor. So it's an interesting approach. So here's that biodistribution across the eye, so mouse, mini pig, NHP, 3 different payloads, pre-recombinase, green fluorescent protein, M-cherry as a red reporter. In all cases, we see robust uptake in the 2 cell types of interest, RPE and photoreceptors. So 3 different payloads, 3 different species, same biodistribution. This gives us a high confidence in the potential to translate to patients. So let me tell you a little bit about the extent of gene editing, and not just the edit itself, but the downstream consequences of that edit. So on the left, you see ARMS. It actually should say not base editor, just sort of Cas9 NLRP3 with the guide. And the top row there is intranasal, and the bottom is intravenous administration. If you do an intranasal administration, 2 doses, you get to 60% editing, which is about a log higher than anything we've seen out there. And on the right, you see the PD impact, right? So there's a pro-inflammatory cytokine reaction. IL-1 beta levels get elevated. You see on the right there, when ARMS are administered, you have almost fully blunted that cytokine reaction. In the bottom, in Kupffer cells, 1 dose, you get to 60% of Kupffer cells edited. And we tried this in an acute liver injury mouse model. And if you introduce the insult of carbon tetrachloride, you'll see a quick spike in liver enzyme levels. ALT is depicted here. Analogous charts behind this with AST and other markers of injury. And again, you see when you administer ARMS, you see a significant reduction in that insult. There's lots of approaches to NLRP3 out there with almost all companies. They're almost always small molecules where you have kind of pan-active and you got to worry about off-target tox. Here you're getting to the cell type of interest. So much more precise. Everything we've talked about so far is the original version of ARMS, where we're leveraging the natural biodistribution of where these particles go. What we're also working on is seeing if you can add a targeting moiety and sort of surface decorate these particles with the intention of directing traffic to get uptake in a very specific cell type. These engagers could be ligands, they could be single-chain variable fragments, they could be nanobodies, et cetera. So we figured we'd start with a very notoriously difficult to transfect population in CD8 positive T cells. So on the left panel, you see our ARM motif there. The payload in this case is mCherry, a red reporter, and the surface engager here is an anti-CD8 single-chain variable fragment. So you see the control and the non-targeted, so original ARMS in the low gray bars showing very little uptake, and then you see the giant red bar showing that we're able to get significant uptake in that one particular cell population. So this is pretty cool. We got pretty excited about that. And we said, all right, well, that's great, but that's a red reporter. Can we actually do anything functional? So we did the analogous experiment, taking ARMS and loading it with a base editor plus a guide. And we ran that same experiment, and lo and behold, we got a very analogous result. So in a mixed population of CD8 positive and negative cells in culture, we showed dramatic uptake in the CD8 positive population. So this is pretty cool. This is sort of our proof of principle, and we're waiting with bated breath. The in vivo version of this experiment is running now. On the back of this success, we're going to try a range of other hard-to-access cells like HSCs, like NK cells, myeloid cells, et cetera. But this opens up a world of possibilities for us. So we're not just limited to the natural biodistribution, but we can start to be proactive and rationally design where we want these things to be targeted. So manufacturing I spoke about. So let me tell you why we're excited about this as a kind of key differentiator within the world of vesicles, or delivery vehicles, I should say. So all of upstream is really just one step. You start with your stable cell line, HEC293. As you remember from the introductory remarks, the cell is kind of doing all the heavy lifting work for you, right? So the cell is doing the loading, the cell is doing the export, and creating for you a sort of population of loaded vesicles. The rest is all separation, right? So separation by size through filtration, separation by charge, some fill and finish. I started my career as a manufacturing engineer. This is pretty easy. This is sort of a log easier than a viral vector process or the complexities of screening associated with lipid nanoparticle process. We've co-published on parts of this with Lonza. We've talked to lots of CDMOs. There's plenty of capacity. You wouldn't need to contemplate an expensive build to get to scale with this. So let me summarize in the last minute or so everything I've said so far. So this is basically the same points I opened with. You have very high loading capacity. So if you imagine overexpressing that protein handle ARDC1, you get a very kind of loaded population of extracellular vesicles. The payload has a lot of flexibility. It could be base editors, it could be proteins, it could be RNAs. As you see these things in transit, they're protected by the natural lipid bilayer, so we don't get any immune reaction, easy to redose. On the recipient cell, target cell side, you see that membrane interaction, so we're avoiding that endocytic pathway, so you get sort of very high levels of functional delivery. We talked a little bit about the natural biodistribution. We talked a little bit about the ability to target tropism. And on the slide previous to this, I talked about the manufacturing process, which is fairly straightforward because you're leaning on the cell to do the hard work. So this is kind of how this all looks when put together. We have a lot of different therapeutic possibilities that we're just on the cusp of unlocking. We think Ocula is a great place to start for us because of the ready access to the back of the eye. So Stargardt's is a great disease for that. We also have 2 NHP studies running in the liver and in the lung, intravenous I should say, and in the lungs. And should those 2 read out as favorably as our first one, we'd be able to very quickly promote a couple other programs to be right behind our Stargardt's program. So it looks like I'm right about time. So thank you very much for your attention. Great to see you. Hope everyone has a great rest of your day. I'm around for a little bit, as are some of my colleagues. If you have any questions, please feel free to ask. Thank you.